Okay, so we're going to talk today about GENCODE, which is the curation of the gene information. And we're also going to talk about the data distribution as well, because these two things we can interconnect each other. So first one is the GENCODE and gene annotation. Before moving on to gene annotation, I'd like to talk about the supplementary table we have for our portfolio, right? So if you open up the portfolio, the supplementary table 2, you will see the information about the genomic and methylation data set associated with this study. So if you look at multi-different tables, so from table the sheet A to sheet I, then you will see whatever the copy number information, RNA expression, mRNA expression, methylation, and mutation, right? So let's look at the RNA expression first, because this is sort of the easy piece. So if you go to table S2D, then you will see the gene expression information for the lung cancer patients from this study, right? And you will see each column's information, right? So the column name ID, column name gene ID, column name ensemble, and the column name C3L0081, C3L0081 something. Differences is N, there's no N. So, I mean, you might have some, questions about that. And from this question, you have to search that information from the supplementary table. There is a answer for that. So you have to do multiple clicking and search the information and make a relation between these and your finding. Okay, so we're not going to moving on to here. We are focusing on this part. So let's guess what is ID, gene ID, and ensemble, right? So first one is ID, and as we can see that this is the column name, right? So this is another data point. So data points start from here. And this is column name, and the column name ID contains the A1BG at the first time. So go to Google, and what is this mean? AIBG, and if you type that, the first one comes with gene. Oh, it looks like gene. Then the first search from GENCODE, then you click that, then you will see that there's a gene name, right? AIPG. So this is actually the gene. And from this search, we can say that the first column indicate the gene name, right? So this is an ID, their column name. Okay, so that's good. So AIBG and AIBG/AS1. What is AS1? It looks like ABG, but it's not. So let's search. We know that this is gene, so we can search that. So you can see GENCODE again, AIBG antisense, right? So now we know that the /AS means antisense, right? Antisense RNA. And some of you might have some question, what is antisense RNA? Search again. There's everything in Google. So if you look at that, antisense RNA refer to antisense transcript, right? At least we know about the antisense because you learned molecular biology. This is the complementary sequence of certain transcript, right? So antisense RNA in here case is the antisense of AIBG gene, right? So there is another gene on the complementary sequence, right? So you don't have to memorize everything. You can search and make relation and inferring from this search, right? Sometimes it's wrong, but mostly it will be correct. And once you do and try again, then your inference is going to be accurate, right? So don't hesitate searching information. Okay, so that's good. So we can say that this is the gene name. And then this is interesting because it's an ID, but there is another ID. So that's called gene ID. So what is gene ID? And search again. So go to here and search it. What is coming? So it's coming from Protein Atlas. It's coming from Ensemble. So let's click one by one. Oh, it looks interesting. And is there any information I'm looking for? Nope. Okay, so go to here. Then this banner. So we can search that. It's not coming here, but at least you can search something similar from here, right? So it has ENSG 000 121410 121410, right? But it's slightly different from here, 0.12. Here is 0.10, right? You know, apart from this, at least this part is same, right? And if you look at here, this part doesn't have 0.10, it has only this part. So you can say that this is another ID. So ID means the identifier, right? For something, right? So we can say that this is the identifier used in Ensemble because it said Ensemble version, right? And it has the same ID here, right? So now you can infer that, okay, so there is a gene name, AIBG, and it has another ID by Ensemble, which is start from ENSG 000 something, right? Okay, so that's good. So let's check our understanding. So let's search some gene you know, right? So which gene you know, right? So any gene name you remember. So at least we know the gene name P53, TP53. So I search from here, right? TP53. Let's search. It's on here, right? And the gene name is ENSG something, 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 right? So there is another gene ID. So at this point, we should have some question why we have to use gene ID rather than gene name, right? So gene name is like, you know, the thing you can actually read, right? So alphabet and you read some information, but here is totally different format, but why it has not a gene ID, right? The answer is when you make gene annotation. So gene annotation means that when the gene newly identified in the scientific field, right? So newly identified means that, you know, the genes you see here, so those genes, there's no point we found this gene all together at the same time. The genes we know at this point, like 60,000 genes in human genome, these genes are consecutively identified by individual researchers. So there is no, you know, one researcher search all the genes. These genes has been identified multiple scientists over a decade, like 2 or 3 decades, right? From 1980s to present, right? So every time you find the gene, you give a name, right? You give a name. Then once the genes has their name, these genes stored in some database, and this database give the ID to this gene, like you know, identifier numbers or passport numbers, right? So if the database called ensemble, this guy give a gene ID by this rule, right? So start from ENSG and 000 something and some numbers, right? But this is only for ensemble. What about the other database? So let's look at here. Go away. Let's look at others. So I don't think this is good example because this is ensemble. So they probably put their ID first, but they have another ID, CSSD. They have an ID, CSSD 12976, and unit prod ID P04217. So if you click this one, they will lead to the database. So CCDS, this information, and this is the unit prod information, right? So why there are so many different databases? Few reason is that each database has their unique point and also their managing body. So Ensemble is run by EMBL, which is European Molecular Biology Laboratory, so one of the largest institute in Europe. So Euro-wise, they operating this institute, and they have the big repository to make gene annotation, right? So they have all the different species, like from human, mouse, zebrafish, the famous one, but they also have like an Angola colobus, bonobo, drill, gelada, or I don't know what that is. Okay, one of the monkey. Okay, not very different from you. And then what about the macaque? Okay, so these are all primates and rodents. And Laura Siberia, I don't know what that is. Alpaca, alpaca is the Q one. And what else we can choose? All cuties here. Cats. Compress this button. Cat, year 2, so cat's genome. And dingo is another branch, right? So all of that information stored in EMBL, right? And what about the CCDS? So CCDS means the consensus CDS protein set. So what is the differences between EMBL and CCDS? Let's look at here, and there must be some information about this, right? So CCDS, so they say their annotation is super good and their annotation is sort of core, which means that their thing is the best one and whatever, blah, blah, blah. They need some convergence between the annotation set, which means that, they will take the lead to make a, consensus annotation sets. And so to this end, they say they started the CCDS project, right? So they say it's high quality, right? Okay, so CCDS, you're good. And what about the UniProt? So what is UniProt? Let's look at the first page. UniProt is world leading high quality, right? So CCDS say they high quality and UniProt say leading high quality, right? But UniProt is actually focused on protein sequence, right? So CCDS focus on gene, right? So UniProt, they say they focus on protein sequence. So all the database has different information. So UniProt, I have lots of fun over the weekend. So this is still holiday, but I'm working a very pity situation. So let's go to one of here. So structure. So if you go to UniProt now, you can see AlphaFold, right? So this is one of the deep learning things. You can turn around. Wow, this is amazing molecule, right? So this is like a circular form, something going between here and here, right? And then you can hover the mouse, then you will see which residues on here. So this is alanine 21, glutamine 20, threonine 19. It's amazing, right? So you can see that. So this is the molecular biology at this stage. So Y first few protein sequence is like this tiny ribbon. I'm pretty sure these things will be removed, right? So my guess is that, okay, I was correct. So here is the signals peptides. So peptides from 1 to 21 is marked as the red, right? 1 to 21. So in structure, it's up to this point, right? So let's look at it again. So this part, I'm pretty sure you learned about the cellular biology, right? So this part 1 to 21, we call the signaling sequence. So when the protein translated, what happened? The translated protein going to Golgi apparatus to be transported, right? So it has to be through the ER. When some of the protein, especially the transmembrane protein, when they go through the ER, it has to be, this sequence will be removed, right? Because this is like a little tag for the transportation. So once these things is done, once these things will be past the ER, then these signaling molecules will be removed. And only this part going to be their place. They're going to be transmembrane or some places, right? So this is the final modification for the protein function. This is the thing you learned from the cellular biology. Otherwise, you can read the textbook. So this is, you can find that information from the database as well. So all the new information we get from here, right? So which functions they're doing. Okay, so they are doing extracellular region, right? So this means that they located in a transmembrane, right? Cytoplasmic vesicles, extracellular matrix, all that information in here too. Okay, so my point is that each database has their own unique points for the gene or protein annotation. And annotation means that, what should we do here? So annotation means that this is very awkward, the English language. I don't think there is a good Korean translator for this, but some people say this is 주석, or some people say this is 참조. But I don't think both Korean words is very correctly delivering meaning for this. So annotation means that once you have certain biological data set, you give a meaning from the external data set. So that case we call annotation, right? So here we call human genome annotation, means that once you have the genome, DNA, then you will say which part is a gene, which part is a regulatory sequence, right? So like we see in ENCODE information, there are many data sets we can use for information. So one most representative method is GENCODE. One is GENCODE and the other is RefSeq, right? So if you click the paper called Hero, I mean, you can see the differences between the GENCODE and the RefSeq because these two things are such a most representative things. So I mean, it's worth reading. But in the short term, I can say that the GENCODE is much comprehensive than RefSeq. So they have all the computational prediction and they have all the experimental prediction, and their prediction is much more sensitive than other methods. So GENCODE has many number of transcripts or the gene information than the RefSeq. So that's why the data has, way larger number of the transcript per gene and also way larger number of translation per gene and also the number of exons or number of genomic coverage is also larger in GENCODE as well. So basically, once you do the biological experiment in these days, you can go either RefSeq or GENCODE. But in my case, I prefer GENCODE because I want to use more comprehensive data set than very finite numbers. Okay, so there was a brief shot of GENCODE data set. So what we have to do is we have to go to GENCODE consortium, the GENCODE page. So if you click the GENCODE page, then you, as we read other databases, we can read the information about GENCODE, right? So what is GENCODE project? This is encyclopedia of gene and gene variant, right? So they have backgrounds. They say NHGRI, which is one of NIH institute, they are doing some research consortium called ENCODE, and they trying to make some functional element in human genome. And after they successfully done something, they want to get some, annotation gene features, and they want to scale up their effort, right? And once they done that, they doing some work, and they set up this title data set, right? Okay, so if you click one of these, then you can see the details. So first one is FNQ, so frequently asked questions. So you can read about the data set. So what does that mean? What is the terminology means? And all the information is stored in here or in a documentation. So you can read that information about the data set. So we're going to go through some of the basics about GENCODE, but you will have the answers over and over. So the documentation will be a good notion to think about the questions. And also, if you look at this information, as we see here, those, gene ID or genetic, the ensemble information, all of that information you're going to use in the data set many times. So why we have to care about that information? Because some of the data set has the ensemble ID, but some of the data set doesn't have that ID information. So in that case, you have to link that information between these two data sets based on the gene ID or the gene names. Or in a certain situation, like, you want to compare the gene expression over whatever the mutation, right? Then you want to merge this gene expression and the mutation together. Then how you can merge that information, right? That case, you can merge by, where are we? We can merge by gene ID, right? So gene ID. Okay, here, right? So we can merge by ensemble ID as well. Then you have some questions, right? So like, I want to merge the information by gene name. Why we have to use the ensemble ID rather than gene name? I can give you two answers. So first one is, most of the situation we have more than two gene names in the data set. So this data set is actually processed. So I think the authors, they remove the duplicated gene name in the data set. But normally, when you have the annotation, when you have the first measurement, the raw data, the data at the first time has the multiple genes in the data set. So sometimes these two gene names appear twice in the data set because sometimes the genes are different, but their name can be same. So that case, you have duplicated information. So that case, you cannot merge that information to the others, right? But ensemble ID is unique to each gene. So unlike gene names, you can actually use the ensemble ID to merge function. So that's one reason. Another reason is that there are some issues in gene name. So this is the gene name. So let's search some of them. So let's go to search March. Let's look at the March. M-A-R-C-H. Okay, so what happened here? So MAPT-IT1, then this gene name is supposed to be March, is supposed to be March 1, but it's automatically it's automatically converted to 1st March, and it's like a date, right? So why that is happen? Because Excel recognized March 1 as a date rather than gene name. To think about how many people use Excel for biology versus non-biology, right? So the answer is obvious, right? So most users, they're actually expecting date when you use March 1, right? So this is automatically conversion here. Then the problem getting bigger. So if let's try this one. So let's read the data set onto here. So we're gonna import this spreadsheet into your RStudio. Then let's look at the data set. So let's search. Okay, let's search by this. Let's search the gene name. Okay, so gene ID, this one, and sample, same as here. Then the gene name is not coming as this. It's coming 43891. What happened? Is it really correct for other gene name too? So let's copy this one and search here. It's not MAPT/1 /IT1, so it's correct. Only happened to here. So these crazy things, why it's happened? The reason is this is stored as this one, right? And if these things imported in R, then it's automatically transit to character, right? And the RStudio thing, this is the POIX. I think it's correct, POIX. This is the format of the date. POSIX, right? So this POSIX form automatically transit into character and with these numbers, right? So what about this one? This is different numbers too, right? 43892. So what we can say, gene name is very fragile to this error. In other words, this gene name always have a chance to make some error in your analysis. So what is the good thing? You can focus your code with using ensemble ID rather than gene name, because these things not going to be contaminated by Excel error. This has to be intact in RStudio. So always ensemble ID or using some official ID from each database, much better situation than using gene name, right? So this is, you have to keep in mind in your portfolio. Okay, so what we want to do is we want to download the data set from we want to fix this data information from the GENCODE. So I want to replace that information from the one I get from the GENCODE. So if you hover the mouse on here, and you can see human mouse and blah, 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 and we go to current release. And human data release version 41, right? and its reference genome is 38, right? So we can say what this meaning. So you go to release history, then you can compare the version. So you see 41, right? This is the thing we get from here. And there is information 21, 19, 10. And there is a genome assembly 38, 38, 37, 37. You know what this means, right? So we learned from the genetics course. Then you click the show all release. You can see 41, 40, 39, 38, 37, and all the orders, right? And you can match that information here too. So what we're going to use, and also here is the statistics, right? So what about the 41 version? How many genes in this data set? There are 60 genes, 60,000 genes, right? And how many protein coding genes? 19,000 genes, right? So, I mean, these numbers, you have to at least know your biology major, right? So some of your friends not working in biology ask you, how many genes coding protein? And you say, nothing, then it looks really dumb. So, I mean, you know at least the number of protein coding genes in our DNA, right? And the long non-coding genes and small non-coding genes and pseudogenes and immunology genes, which is, you know, important for the COVID-19 infections or your development. And how many transcript in our genes, right? So number of genes is 61,000, but number of transcript is the 251,000. You know why transcript has more numbers than genes? You know about the region, right? Because gene has multiple isoforms. Why we have many different isoforms? Because it has a advantage in our development, in our activities, in our homeostasis. This is the things we get from the evolution, and all the biology has complexity to make a redundant we have a redundancy over the environmental exposures or developmental context, right? So human gets complicated by these isoforms and splicing. So we have many numbers, and also protein coding transcript has also many numbers as well. But this transcript is not only full-length protein coding transcript, some of it has partial length, some of it has nonsense mediate decay transcript. You know, you learned about that in molecular biology. Some of the transcript doesn't, not really under translation. It's totally removed by nonsense mediate decay machinery, right? Because it's short, and if that is not going to be disappeared, will be very deleterious in your cells or in your development. So some of the transcripts, which are very shorter than the full transcript, it's all under removal by nonsense mediate decay process. Okay, so you can see further statistics from here. Okay, so what you're going to do is you can download one of the file. I'd recommend the basic gene annotation. And what is the differences between basic and comprehensive or long non-coding RNA? It's not about the price. It's not about the packages. It's about the different compositions. So basic annotation, you only have basic gene annotation on the reference chromosome, right? So you might have some questions like, what is the non-reference chromosome, right? And non-reference chromosome probably appeared in here because these two things are contradictory to each other. So if you see that reference chromosome, but not reference chromosome things is scaffold assembly patches or alternative loci, right? These things are non-reference chromosomes, right? And if you click GTF file, if you click, then that will be appear like this, and you can save. And this file, it's about 30 megabytes, so it's not that small, so it might have some time, but Once you loaded that, then you can move to your working folder. So what you have to do once you download, then you have to install. Okay, so I mean, this is the tutorial you have to submit some point. I might make some change, but I mean, you can follow that. But one note is this is the version 31 and you are using version 41. So it's different because the, the version of when I make this, it was 31 at the latest. So this can be changed because some of students might ask some questions. Okay, so you can read the data set like this. So you can read the data set, use the tidyverse, library, tidyverse, then it's 41. Okay, it may take some time. I don't think it's working correctly. It's working correctly, right? So I can read some 9 columns and it has chromosome. It has some weird information on here and weird information in here and some numbers and some information too, right? So how can we get that information? So it's all in the internet. So if you go to GENCODE, then you can go away. You can go to documentation, maybe data format. Okay, so what we just downloaded, we download the GTF file. So we can search. Okay, so this is the format description. And this is the tab separated GTF column. So first column, what is meaning is the chromosome. And second column is the annotation source, right? So it has either ensemble or Havana, right? So I can tell you a bit of background. So GENCODE is the joint effort by two reference database. The first one is ensemble, which is the one we just saw. So this is the ensemble, right? So European Institute. And they also have Havana, right? What is Havana? Havana is a beautiful place. Havana is music, but Havana is not just music. This is one of the Cubanian city, one of the historical city in Spanish. In Spanish, the dominancy period. It's in Cuba. It's just under the Mexico. Okay, so but this Havana is not the same one as this one. Havana data. Oh, who is this person? Havana. Okay, so this is another database, the manual gene annotation by Havana, right? So this is another effort. Go away. This is another effort by Ensemble as well. So Havana is doing manual gene annotation. So manually, you can examine what genes are annotated in a database. So ensemble annotation is automatic process, but Havana is by manual curation, which means that people see the data and look at the annotation information. Okay, so these two sources in GTF. So my curious is how many done by Havana and how many done by Ensemble. So Havana is how many? The ratio, I mean, it's almost like 12 times bigger by Havana, right? And the feature type is the gene, transcript, exon, CDS, UTR, start codon, stop codon, and selenocysteine, right? Then you can see that there are information about gene, there are information about transcript, there are information about exon, CDS, right? So you can see a hierarchy, right? Gene is the superset. Under the gene, it has transcript. Under the transcript, as you learned from the molecular biology, but some of you might be forget about that. So let's look at together. So CDS, exon and transcript. So this is one. So exon, 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 intron, intron, and UTR, right? And once these things all chopped away by splicing, then exons are all going to be CDS, right? So all that information is stored in here: exon, CDS, UTR, start and stop codon. Start and stop codon on here, right? And selenocysteine is, what is this? This is another form of the non-canonical amino acid, right? So another amino acid you will use for the metabolism, right? So these things also stored in GENCODE information too. So if you want to know about the gene information, you have to subset the gene from this data set. So let's clarify what we have to do. So we see that the March 1 information is totally correct in this data set. So we want to fix that information by this ensemble ID and gene ID. So how can we do that? So damn it, I have to hit this file again. Okay, I will put M on here. So I will import the data set of this, right? And I get the data set about the GENCODE. So what I want to do is I want to filter. So what was the information? So that was the X3. X3 equal gene. Then I want to get the information about gene ID and gene name. So it's in an X9. Okay, so there has to be X1. Okay, so X9 column has gene ID, gene type, blah, blah, blah. But that is super, complicated. So not many people wants to chop this string into the new column. So, I mean, there's not really easy way. So what I want to do is I want to use other packages to load the GTF file, which is Artrack layer. So, I mean, this package is not installed in your computer. So let's install this one together. I mean, sorry, by yourself. Then let's load this data set. So let's load the data set together. Okay, it might take some time because it's a huge file. Okay, so it's done. And let's check the data set together. So what is this D? So it looks like something different, large G range. They say as the class is not data frame, it's not matrix, it's not list, it's called G range. So this is the class type by genomic range packages. So this is very specific for the genomic data set, but we can actually ignore this data set for now. So if you look at this, it looks like data frame, but it's slightly different. But we're not going to use this one. So let's transform that information like this. So I forced to convert the information into the data frame. And surprisingly, it's working because there are, I mean, similarity about the data formation. So as you can see here, the X9 column, it's all separated out by its features information. So gene type and gene ID, whatever, where are you? Gene name, something, something, right? So what I want to do is I want to filter type because the type is gene here, right? So I want to filter gene. Then I want to filter what I have to do. Select gene name and gene ID. I think that is enough. Okay, let's do this one together. Then there will be a like this, right? So I want to use this gene ID and I want to merge the information to fix this information, because if I merge this information and this one together, sorry, let's look at this one together. If I merge this information and this information, then I can fix the gene name on here with this gene name. So what I have to do is this gene ID probably same as this one, but I'm not sure whether they have this point some numbers. This is the minor numbers for the gene ID. So sometimes this is not matching to matching between the versions. So we have to ignore these numbers. Then we can merge that information. And then I will ignore this ID. How can we do that? We can just remove this column. Then we can merge with this one. So how can we try that? So first one is we have to ignore these minor versions. So is there a way to do that? Damn it. So file track layer. Let's look at the manual together. So that's probably this one. Nope, I don't think so. Is this working? I don't think so. In that case, we can search the internet, import, ignore minor version. Not really appeared at the first search. I don't think it's working. Ignore. Ignore gene ID. Interesting. Track layer, ignore. Data import. Okay, so there's a manual page for that. No, this is not. Maybe this is working. Version 3. I don't think it's working on here too. So that case we can remove by manually. So how can we do this? So that's my job. So let's look at the mutation function. Then I want to make a gene ID 2 to call one data frame. The gene ID. That's 1. Is this working? Okay, so it's working. So it's too quick, right? So you have to press the button in YouTube, or there must be a way. I suggest the way in here. Did I? I think so. Yeah, I think I put some similar things on here. Otherwise, you can go to Slack channel. Okay, so what I have to do is I can merge between this and this data frame. So I want to first make a simple version first. So gene ID. Okay, so add the one. Okay, that's good. So what we want to do is we want to do merge the D1 and M and Y. So the merge function is you merge data frame, the first data frame and second data frame. So if you have the two data frame, and if you want to merge between these two, then you can use the merge function. And the first data frames column, you're gonna use GENCODE, right? And the second column is you use ensemble, right? Because these are the same one, right? So you can use like this. So you say it's going to be E. How it looks. Okay, so GENCODE here and GENCODE.y, because these are duplicate column numbers, it has another one. I mean, it looks ugly, so we're going to remove that. And then the gene name, now it's fixed, and ID is this one. So this and this column's not gonna be used anymore. So how can we remove that? PT. So, I mean, before removing that, we have to search that information, whether it has the correct gene name now. Okay, now we have the correct name. Its name is MTARC1. It's not March anymore because the people now they fixed the gene name because it's the exact crash. So probably from GENCODE version 40 or 41, they start fixing this name. But anyway, now we have the correct gene name. Then you can use for the your merge. So let's look at the merge function more tidy way. So we want to remove the column name is ID and gene ID, right? So ID and gene ID. So we're going to remove these two things because these two things not going to use in your analysis. So you remove this one, then you can do merge. Oops, go away. Then now you can see that gene ID, what? You have to do this one. So you have to put minus. Then it's going to be go away. Okay, so it has gene ID and gene name. So this is what we want, right? So this is one example. We are looking at the merge function. So why we have to care about that information in your data set. So let's think about the situation. You get a information about the gene expression. So let's assume that you want to compare the expression of AIBG1's expression between the female lung cancer patients versus male lung cancer patients. Then you have to get the information about these samples, right? And information about these samples, where we can find it, that information, it's in the supplementary table 1, right? So if you go to supplementary table 1, then you can see which sample ID means which, right? So it has this sample ID, it's a non-smoker, and it's a female, right? So you can get information in here. So if you want to merge that information, then you can use that data frame and use this data frame. Then you can merge that information for your analysis, right? So in that case, you can also use the information as well. Okay, so that was the brief introduction about the merge. And let's look at the information about the gene mutations too. So this is the table S21, S2I, and it has the ENSG information, which is the gene information. Where are we? So here, gene information, right? But interestingly, it has ENST information too, which is in here, right? So it's annotated by transcript, right? So why mutation has the information about transcript as well? So if you look at the RNA expression, it only has the gene ID, right? There is no transcript ID, but mutation has the transcript ID. Why, right? So you have to think about that. So if you come up with the good answers, and let's talk further and let's discuss further in Slack channel, because this is such an important question for your exercise. So let's think about that together. Okay, so that was all about that GENCODE information, and I think it's matter of time. So we have to keep this one for today, and let's move on to data visualization in other class time. Okay, so that was all about the things, and have the exercise on this data set, and also keep thinking about how you integrate that information into your portfolio. That would be super useful. Okay, see you next time.